Welcome to The Explainer. Today, we're going to dive into one of biology's most fundamental concepts, but also one of its most misunderstood, the gene. It's a word we all use, but its real story, well, it's a scientific epic full of some amazing twists and turns.
You've definitely seen this picture before, right? Probably back in a high school biology class. It's that classic, simple idea of genes just lined up neatly on a chromosome. Each one is like a little bead with its own specific job. It's clean, it's tidy, and it feels really intuitive. Okay, let's really get into this, because the real story of the gene is just so much more complex, more interconnected, and honestly, way more fascinating than that simple model that's on. That neat little picture, it's been unraveling for decades. Now, before we tear down that old picture, we should probably appreciate why it was so powerful to begin with. You see, this simple story of the gene wasn't just some random guess. It was a hard-won concept that, for almost a century, was the central unifying idea in all of genetics. For decades, science was building up this really powerful unified idea. It all started with Gregor Mendel and his abstract factors for pea plant traits. Then you had Thomas Morgan's famous work with fruit flies, which gave those factors a physical home right on the chromosomes. And all of this led to the one gene, one enzyme idea, which gave each gene a really clear, specific job to do. And here's the absolute key takeaway from that whole period. The classical gene was seen as this one, single, unified thing that did everything. It was the indivisible particle passed down to your kids, it was the blueprint for a specific protein, and it was the exact spot that changed during a mutation. One entity, three core jobs. Simple, elegant, and incredibly powerful. You'd think that the discovery of DNA's structure back in 1953 would have been the final triumphant confirmation of this beautiful, simple idea. But, kind of ironically, the more we started to learn about the molecule of life, the more that classic gene concept really started to fall apart. So first up, the idea of the gene as an indivisible particle, that got completely shattered. Molecular biology showed us that when sperm and eggs are made, a process called recombination literally breaks functional genes apart and shuffles the pieces around. The only thing that's truly indivisible is a single DNA letter, not the whole gene. Then, there was the idea of a gene as this single continuous recipe. Well, that got dismantled too. Scientists discovered that a ton of genes are actually interrupted by these long stretches of non-coding DNA called introns. So the cell has to copy the whole thing, and then literally snip out all the nonsense parts and stitch the meaningful bits back together. The recipe is broken into pieces right from the start. And finally, the whole beads on a string model just collapsed under the weight of this incredibly complex regulatory system. We learned that genes are not independent actors at all. They're a part of a huge network, getting switched on and off by other pieces of DNA. Some genes even physically overlap or are found inside other genes. It's really less like a string of beads and way more like a circuit board. So with all its core beliefs broken, indivisible inheritance, continuous function, and independent action, that single, simple definition of a gene just didn't hold up anymore. The concept had completely fragmented. And this forced a huge shift in thinking. Biologists and philosophers realized they had to move away from the frustrating question, what is a gene, and start asking a much more practical and frankly more powerful question, what does a gene do? To answer that, we can turn to this really powerful philosophical tool called difference-making. The idea is actually pretty simple. A cause is just something that, if it had been different, would have made the effect different. So, for example, if changing a specific bit of your DNA is the difference between having blue eyes and brown eyes, then that DNA sequence is a difference maker for eye color. It lets us pinpoint specific causes in a really complex system.
This whole concept of difference making also helps us distinguish between different kinds of causes. Think about it like this. You're reading the news. The oxygen in the room is a cause of you forming a belief. I mean, without it, you couldn't think. But it has really low specificity. The words on the page, on the other hand, are a highly specific cause. You change the words, and you precisely change the belief. Genes are often like the words on the page. They're highly specific difference makers in the story of our biology.
This framework really helps us use our metaphors for DNA way more precisely. So as you can see here, the idea of a genetic code is totally legitimate when we're talking about the specific mapping of DNA to amino acids. And the genetic program is a pretty good metaphor for the logic of gene regulation. But these metaphors get really misleading when we stretch them too far, like saying a gene codes for a complex trait like intelligence. The reality is that DNA is both a library of specificities, that's the code, and a part of the cell's control system, that's the program.
Okay, so if genes are these specific difference makers tucked inside a complex cellular network, how does this new view fit into the grander scheme of things, into evolution?
And this leads us to the famous selfish gene perspective, made popular by Richard Dawkins. It's such a powerful idea that totally reframes evolution. It treats genes, not us organisms, as these central players, and our bodies are just the vehicles they build to copy themselves.
But according to philosophers like Peter Godfrey Smith, this powerful idea isn't a literal description of reality. It's what's known as a scientific idealization, which is basically a deliberate simplification. It treats genes as if they were those old-fashioned particle-like actors because, well, doing that creates a really useful model.
And this is a brilliant illustration of its use. The selfish gene model works perfectly when you're zoomed way out, looking at evolution on a macro scale, tracking gene frequencies in a population over millions of years. But it becomes a pretty misleading oversimplification when you zoom in to the micro scale and look at how an actual highly interactive genome really works inside a cell.
Now, that's not to say that truly selfish genetic elements don't exist. They absolutely do. Things like jumping genes can copy and paste themselves all over the genome. But, and this is the important part, they are a special case. They're an exception that actually proves the rule of a largely cooperative genome. They're a derivative possibility, not the main way that all genes act. So, where does all this leave us? Let's try to piece it all together and form a new, more accurate biography of the gene. We've been on quite a journey here. We started with that simple story of a particle-like gene. We watched that whole concept fragment as we learned more about the messy reality of DNA. Then, we rebuilt our understanding by focusing on what genes actually do as specific difference makers. And finally, we placed that role within the bigger context of evolution. So, the ultimate takeaway is this. The gene isn't a puppet master pulling all the strings. It's an essential but fully integrated component. It's both a library of highly specific information and a part of the complex control system that decides when and how that information gets used, all working within the incredible machinery of the cell. This whole journey, from a simple particle to a complex interactive process, it fundamentally changes how we should think about the instructions that build life. I mean, if there's no single simple blueprint, but instead a dynamic, responsive system, what does that really mean for who and what we are? That's the question this new biography of the gene leaves us with.